All right, so we're going to start with carbohydrates today. We've talked about polymers and monomers. We've talked about dehydration synthesis and how we put together those monomers to make polymers. So now we need to discuss what are our categories of polymers or macromolecules that biological organisms require. And our first group are carbohydrates. And carbohydrates have some very specific characteristics. And in this section, we're going to talk about the macromolecules. We're going to focus on the characteristics of them and their uses in our bodies. So carbohydrates are easy to spot, easy to pick out if you're looking at formulas because they only have carbon, hydrogen, and oxygen in them, nothing else, no nitrogen, no phosphorus, no sulfur, just CH and O. And even more telling about a carbohydrate is that they have a specific ratio. Their ratio is 1 carbon to 2 hydrogen to 1 oxygen, giving them the empirical formula of CH2O. And of course, that will vary depending upon, are you talking about a monosaccharide? If it's a monosaccharide, it would be C6H12O6. If it's a disaccharide, it would be double that minus 1 water molecule. If it's a polysaccharide, it would be how many ever monomers we have there minus a water for every bond that holds 2 monomers together. So what do we need carbohydrates for? Well, we need them for energy primarily. They have a lot of energy stored in those carbon-hydrogen bonds. The hydrogen with the 1 electron that it has is something that when we break it off, we can use those electrons, if you remember, in cell respiration, right? We pump the hydrogen electrons or the hydrogen atoms across the membrane, stripping the electrons and using the electrons in electron transport chains. So they are very useful for energy production and storage. Examples again, simple sugars like we know of, glucose, fructose, etc. Starches, which are your complex polysaccharides. So to look at the different groups here, we have monosaccharides to begin with. It is our simplest carbohydrate, fast energy. There are only 6 carbons in it, and our simple sugars play important roles in biological systems. Glucose, again with the formula C6H12O6, is the most common form. It's like the universal food, if you will, universal energy molecule that all living things can process and break down and turn into ATP. We talked about isomers also in this chapter, and fructose is an isomer of glucose. It's a structural isomer, so its formula is the same, C6H12O6, but structurally it is different, which gives it different properties, so it's a different kind of sugar. Galactose is a stereoisomer of glucose. So we talked about stereoisomers, meaning their side groups are just mirror images of each other. So galactose and glucose, also another example of isomers. Why they are different, how do they have different jobs, or what do they use differently for in our bodies? It depends on the enzymes that we need to break down those sugars to get energy out of them. Enzymes are specific. They can only work on certain molecules. So the enzymes that work on glucose do not work on fructose or galactose because they can tell a difference between the isomers in this 6-carbon sugar. So our enzymes are very specific, and their active sites, which are the locations on them that the sugars fit into to be processed, are specific and specific for the structure. And so again, they can differentiate between a structural isomer of fructose and glucose or the stereoisomers of galactose and glucose. You can look here, we can see what structural isomers look like. Sometimes they're in a chain, sometimes they're in a ring. You can have something called alpha glucose or beta glucose, which is an important difference in the production of starch versus glycogen. And the difference here is just where that hydroxyl group is at. In alpha glucose, that hydroxyl group is below the 1' carbon. In the beta glucose, the hydroxyl group is above the 1' carbon, and that is an important difference between the two as far as how our enzymes can break them down. So you see glucose in the middle, our structural isomer to the left, which is fructose, the stereoisomer to the right, and the difference there is just the flipping of the side chains, whereas with the structural isomer, you have differences in where the carbonyl groups are at. So just looking at how we have different isomers between our simple monosaccharides. So what happens when you take two monosaccharides and remove a water molecule through dehydration synthesis? You get a disaccharide. So down below, it's showing you the process where water is removed, and that creates a bond between an alpha glucose and a fructose, creating sucrose. Disaccharides are important. We use them for, a lot of it is like sugar transport. They are relatively quick to break down, not as fast as glucose, but energy storage is another purpose for them. Really, monosaccharides and disaccharides are just about energy purposes. So sucrose, lactose, which is milk sugar, right? Maltose, malt sugar. Ose, did you notice the end of every sugar molecule when you're a monosaccharide and a disaccharide, you always end in ose. That's not true for polysaccharides, but for the first two categories it is. So that takes us to polysaccharides. Polysaccharides, many saccharides, right? Many monosaccharides linked together, again, through that dehydration synthesis process, used for two purposes. First one is energy storage. Polysaccharides, when they're made up of so many monosaccharides, are good for long-term energy storage. So plants will store their energy for long-term as starch, which is what we are eating when we eat a carrot, any kind of root vegetable, any kind of, well, really any part of the plant besides like the leafy green part. If you're eating a stem or a root, there's starch stored there. And that's how plants store their excess glucose that they make during photosynthesis. Now, animals, when we eat food and we store our excess sugar, you know, if you can't use it immediately and we need to store it, we store it primarily as glycogen in our liver. So where plants store their sugar as starch in either their stems or their roots for the most part, animals store their excess sugar as glycogen in our liver. The second purpose of a polysaccharide is structure. We don't necessarily use polysaccharides for structural purposes too much, but plants and other organisms do. So plants use cellulose, which is a polysaccharide, different than starch, but they use it for structure. And so that's what gives secondary growth possibilities in plants, right? Helping them grow taller and wider. Arthropods and fungi use a form of polysaccharide called chitin. And so it is another type of polysaccharide. It's just a little bit different in its makeup than cellulose. And so here are some differences. When you're looking at these images, right, you can look at the differences between, what we're trying to look at here is a difference between starch and glycogen, and the main difference is where the bonds are at. We can break down glycogen for energy, but we can't break down cellulose for energy, and it's all about where the bonds are at between the glucose molecule. Same here, we're seeing bonds between our monosaccharides. Some of them, they can be broken down and some cannot.